morning, everybody. My name is Cindy Shimoleski, and some of you may know me as the myeloma teacher, and I am here with the patient story today. And today we're going to be talking about the latest and greatest things that are happening in multiple myeloma. For those of you that don't know me, I've been living with myeloma since 2008. And more recently, I became a research advocate in the multiple myeloma community. With us today, we have Dr. Usmani from Memorial Sloan Kettering, and I'll let Dr. Usmani take a few minutes to introduce himself.
Thank you so much, Cindy. It's a pleasure to be here chatting with you. I'm the chief of the myeloma service at Memorial Sloan Kettering Cancer Center in New York. And I've been involved with myeloma research, both on the clinical and translational side of things, for well over a decade. And I'm looking forward to chatting with you about so many exciting things that we have in the field and how things are moving forward for our myeloma patients.
Great. So there's been some meetings going on. There's ASCO and EHA, which I actually got to see you at EHA this year. And the IMW is coming up. So there's a lot of things happening in multiple myeloma. So let's start talking maybe about the CAR-Ts, because CAR-Ts have been around for a while. And can we talk about, I think there are 2 approved CAR-Ts. Can you talk a little about them and who they're for? And
certainly. So CAR T cell therapies, have been around for a while. As you said, they started early on in another disease called ALL in children, actually. But the ones that are developed in or have been developed in myeloma are targeting a specific surface protein on the cancer or myeloma cells called BCMA. And there are 2 that are FDA approved for patients who have had 4 or more prior lines of treatment, including a proteasome inhibitor, immunomodulatory drug, as well as anti-CD38 monoclonal antibodies. So really people who have had a lot of prior therapy. And those 2 products are called ide-cel and cilta-cel. And, they both target BCMA, but they are unique products. So, the general side effects, are similar, but tend to happen at different time points. And we can talk a little bit more about this. But both of the products actually show, good clinical activity. And we can talk a bit more about that as well.
good. So these CAR T cells, though, they have to be given in like a center of excellence. Is that correct? I can't just go to my doctor and say I want some CAR T treatment.
Yeah, so these are, cell therapy products. So they need to be given at a center that does transplant and cell therapies. So many of our myeloma patients do go for a transplant evaluation. And end up getting a stem cell transplant during the early course of their treatment. So the process is kind of similar. Your doctor is going to refer you to a transplant center. They will do the evaluation and then, get you those commercial products. Typically what happens is, when you're getting a stem cell transplant, you go through a process of getting growth factor shots and then getting the stem cells collected from your bloodstream through a central venous access, typically. In CAR T cell therapy, we are collecting T cells from the patient's peripheral blood, but you don't need growth factor shots. So you can get those T cells collected just off of the peripheral bloodstream.
Okay, good. Now, there are some unique side effects to CAR T cell therapy that maybe myeloma patients haven't experienced before. Can we talk a little bit about them?
Certainly. So the way that the process works is, when you get these T cells from the patients, you have to send them off, to the specific, company that's manufacturing them for the manufacturing process. And what happens is, in a layman's way, the patient's T cells are taught to recognize the cancer cell, and they're grown outside, the patient's body, in the lab, essentially, so that production and expansion takes, 3 to 4 weeks. When that product is ready, it's shipped back to the center and patients get a certain, lymphodepleting chemotherapy for 3 days before they get that product infused through the veins. And those T cells are active. They know how to recognize the cancer cells, through the surface marker BCMA, and they're going to expand. So there's an expansion that happens, in the patient's bloodstream, and these cells are growing, and they're trying to go after the myeloma cells. It's like you are introducing, an inflammatory response, in the patient. So the side effects are like the patient's body is going through, an inflammatory process. So the two common side effects are what's called cytokine release syndrome. These immune cells are releasing certain chemicals that help them, enable them to go after the target. And due to that, patients can feel, fevers, hypotension, as if, they were infected with something. So it's that tends to happen with ide-cel within the first 2 days. With cilta-cel, it tends to happen with a delay of 5 to 6 days typically. And we manage those kind of side effects by giving antipyretics, fluids, sometimes, steroids or an antibody called tocilizumab is given to patients. But for the most part, these side effects are grade 1 or 2, so they're kind of low grade and are managed, with those kind of medications. The other side effect that can happen because these T cells are active is, what we call neurologic side effects. So, patients can, sometimes patients can feel, loopy or have neuropathy. Very rarely, patients can have, more serious neurologic side effects such as seizures. Those tend to happen in very low proportion of patients. Neurotoxicity tends to happen more commonly with with ALL CAR T cells rather than the myeloma CAR T cells.
Are there any biomarkers that might predict who might have a greater tendency for severe neurotoxicity, or is that just up in the air?
So what we've observed is patients who have a higher burden of disease at the time of getting CAR T cell therapy can be at a higher chance of developing these kind of side effects. One can develop, CRS and neurotoxicity independent of each other. So it's not that they come in as a package, so in fact, CRS is more common. Neurotoxicity tends to happen in, less than 10% of the patients. So CRS tends to happen in almost all the patients to some degree. That there aren't typically, specific markers that we can follow without, that are specific for CRS or neurotoxicity. There's certain markers that can give you a hint that there is a heightened sense of inflammation. So there are certain tests like, serum ferritin levels, or, you can have CRP elevations in patients. So we sometimes tend to follow some of those labs, but they are, they're not predictive. They're just telling you that something is going on. Just be careful, but they're not very predictive.
Okay, that sounds good. So the currently approved CAR Ts are for probably more heavily pretreated patients who've exhausted many of the options. Are there any CAR Ts trials that have been reported on for more newly diagnosed or first relapse? Are they coming closer to the beginning?
There are 2 randomized phase 3 studies that have, looked at comparing, each of these CAR Ts, both ide-cel as well as cilta-cel, in 1 to 3 prior lines of treatment, comparing them to the standard of care triplet regimens. In fact, this interview is, fortuitous because BMS just announced, I think 3 or 4 days ago, that their phase 3 trial in 1 to 3 prior lines of treatment is coming out positive. Now this was a company announcement, so we haven't seen the data, but that is good news, for our patients. So, I'm keen to hearing more about this. We do have smaller studies, smaller cohort studies that have reported out experience in 1 to 3 prior lines of early relapse patients with very high response rates of, 80, 90% with cilta-cel. In fact, well over 90% with cilta-cel. So we just need, more update data. Those are, looking promising. I think the key is, the process that I explained about, CAR T cell therapies. It takes a while for that production time. And, so there are certain bottlenecks to the process. So the big question is, how applicable, this technology will be to all patients, that we treat.
Yeah, and because of all that, there's that bridging therapy and things like that. What about these allocarts? Are they going anywhere? What are they? Can you explain? And they seemed a little exciting.
Yeah, so the idea of alloCAR Ts is, you can take T cells from a donor, but then knock out certain surface markers to make those T cells, more benign, so that the recipient's immune system is not recognizing them as the enemy, and they're not recognizing the recipient's immune system as the enemy. The advantage of having allo T cells utilized to manufacture CARs is you can have one donor and make a product that can be given to 100 different patients instead of taking the patient's own T cells. And those hundreds of products can be given in a very short duration of time. So essentially, if I see a patient today and I have an allo CAR T available, that patient can technically get that product 3 or 4 days from now. They can start getting lymphodepleting chemotherapy and they can get that product very quickly. Whereas if I were to see a patient today and try to collect their T cells, even with everything kind of aligning, it might take 5 to 6 weeks for them, from today to get that product. So that's the, major advantage of allo CAR T products, but they are, they're way behind in terms of clinical development and seeing the light of day.
We have any efficacy data? Are they working or?
Yeah, they are working. So we have seen some efficacy data. There's an allogene CAR product. In fact, Sham Myeloma from my program had presented those data last year, showing, about 60% of the patients responding to that allo CAR product. Now the follow-up is short. So we know that these patients too get CRS and neurologic side effects, but the follow-up was only about 3 or 4 months. So we need longer follow-up to see, is this activity long-lasting or just, a short duration activity? And what kind of long-term side effects do we get from using this strategy?
Good. And one more question on CAR T cells before we move on. Right now, the approved ones go after BCMA. Are there any CAR T cell therapies that are looking at different targets on the myeloma cell?
Yeah, so another target is called GPRC5D. Again, that was a homegrown, CAR T at MSK that Dr. Myeloma had presented, showing good activity in patients who have had prior BCMA CAR Ts and BCMA therapies as well. The response rates, again, in high 60, 70% range. Looking at, the various cell doses that the patients received. And that CAR T is now, being developed, by BMS. So there are other programs that are utilizing that CAR T. We also have some very interesting, dual CAR T, BCMA and CD19 CAR T data that was presented by colleagues from China at the ASCO and EHA meeting. Showing high response rates. And the cool thing about that platform was, the short production time. So that product was being manufactured within 48 hours. So what we're doing is, what we're seeing is the new platforms are actually reducing the production time, and that will be, a good challenger to the allo CAR T strategy. If we are able to shorten that time, then, there might not be as big an advantage for an allo CAR, compared to patients' own T cells.
Can we talk a little about Talquetamab? I think it's already approved in Europe, isn't it? Or
it got a favorable, response from EMEA, but it's not yet approved, and we're waiting to hear from the FDA. But certainly, I've been involved with Talquetamab from, the very beginning. In fact, the first patient who was ever treated in the world was my patient. So I've been with that program from cohort 1, patient 1. I think CAR T cell therapies are very effective, but bispecifics, are therapies that will change how we manage patients in the community at large. And they actually might challenge, CAR T cell therapies, because, they're an off-the-shelf option, giving, high response rates of, again, 60-70% in relapse refractory myelomas, very similar patient population to CAR T cell therapies, and it's given subcutaneously. Now, cytokine release syndrome is something that we see with the bispecific antibodies as well, and that's something that we have to watch out for. The concept of this technology is very interesting. So, you're going to hear TCE or T cell engager as a terminology. You're going to hear bispecific antibody as a terminology. The concept is the same. One part of that molecule recognizes the surface marker on myeloma cells. It could be anything. It could be BCMA, it could be GPRC5D or something else. And the other part of that molecule recognizes the T cell. So, there is, surface marker CD3 that it recognizes. So, so the difference between T cell engager and bispecific antibody is the structure of that molecule. If it's a whole antibody, then you call it a bispecific antibody. If it's only a fragment of an antibody, you call it a T cell engager. And different companies and platforms have different molecule structures that are proprietary. So the names are going to be different. So long explanation, but a lot of excitement about that technology. Talquetamab is the front runner. We might hear some good news about it later this year in the US and Europe. And we're all really excited about that platform.
Yeah. I guess for patients, it's exciting because it doesn't involve having to probably relocate like the CAR T cells. You have to go to a CAR T center and spend time away, maybe from your home and family. Would the bispecifics be something that could be given maybe at a community hospital, or is this something that you need to go away for too?
Well, I think there will be a learning curve for our community physicians because giving bispecifics does require, some education and comfort in managing cytokine release syndrome. So, it's quite possible that they might ask the academic centers to give the first cycle of treatment. Patients do need to be monitored during that first cycle as they're going through the various step-up dosing to full dose for that cytokine release syndrome. And because our community colleagues don't see myeloma as frequently as some of the other solid tumors, they might ask us to give that first cycle. But once patients are beyond that first cycle, it's like, getting any subcutaneous treatment. So, the short answer is yes, it will be possible for patients to receive this treatment out in the community. But I don't see this happening during the first year of the FDA approval. It will be, a steep learning curve for our community docs.
Okay. But usually after the first cycle or two, the CRS and the neurotoxicity seems to go away, and it's just, is that?
Yeah, so the likelihood of having those side effects is during the first, 3 or 4 doses, so that first month. And it's really about again, engaging the T cells, right? So once the T cells are active and they know exactly what to do, and they're fine with the job assigned to them. It's that early part where you're trying to say, hey guys, go and kill the myeloma cells, and they start, they get revved up and release those cytokines, and once that process is done, the risk goes down to neglect.
Like for Talquetamab, is the infusion like weekly, monthly? How often would I need to get this sub-Q injection?
So the sub-Q injection is on a weekly basis. The first 10 days, the step-up dosing is given every fourth day for the first 2 doses before you get the full dose. So I think that's how it was done in the studies. I would be surprised if the FDA approved label says something different. But again, we will know what the FDA says later this year.
Okay, and what about the efficacy? How does it compare to CAR Ts?
So response rates are around 63, 64%, which is very high. And the median duration of response is, or I should say median PFS is about a year. So almost 12 months. So if you recall, the approvals for carfilzomib and pomalidomide and even daratumumab, the response rates were 25 to 30% for each of those products. So and the PFS for those products was about 4 months. So that gives you the context that, you have this bispecific antibody. These are patients who have progressed on treatments like carfilzomib, pomalidomide, and daratumumab, and you're still seeing these high response rates and median PFS. In fact, the median PFS is similar to what was reported for ide-cel. Ide-cel's PFS was less than a year, and the response rates were over 70%, but median PFS was in a similar range. So very interesting data, and very encouraging data for our patients who have had triple class refractory disease.
Talquetamab, okay. And does that go after the same target or a different target, or what about?
So it's in the same patient population, but the target is GPRC5D. So GPRC5D is a unique target to plasma cells. Talquetamab appears to be active. Response rates are up in the 60s% as well. We don't have the PFS data because the development of that particular bispecific is a little behind Talquetamab. But we're seeing activity of Talquetamab even in patients who have had BCMA-directed treatments, including CAR Ts and bispecifics. So it's looking very promising.
That's good to hear. And there's many others, right? There's not just these two. There's a lot of bispecifics out there. Are they all doing quite as well?
Yeah, so, for BCMA-directed bispecifics, there are several options. So elranatamab, is very active. ABBV, 838 is very active. There's a Regeneron bispecific. There's a PMS Celgene T-cell engager that's targeting BCMA as well. All of them are showing very similar kind of activity. I think there are some differences in the safety profile, percentage of patients getting cytokine release syndrome when they're getting it. So I think some of those issues will be the distinguishing factors. And there are some that are given less frequently. So, I think the AbbVie bispecific, which was actually developed by, a smaller company called TeneoBio, it's given every 3 weeks. So I think there will be some nuances or differences between, these BCMA-directed bispecifics. And then for GPRC5D, we only have one, but there are others that are in early development. And then we have another target called FcRH5. There's an antibody called cevostamab, which appears to be active. Response rates of about 50% in patients who have, relapsed refractory myeloma beyond 4 prior lines of treatment. So it's good to know that there are other targets where BCM, beyond BCMA and even GPRC5D, where we can see, good clinical activity in patients.
And as a patient, that makes me feel good. Even though some people say, well, you might be able to use another BCMA-directed therapy. For me, I would like to have another target in my back pocket. So keep developing those targets there. There were these TRIM trials, TRIMM, TRIM trials. So now they're not only using the bispecifics, but they're combining them with things. Can you talk a little bit about those TRIM trials?
Yeah, so once the efficacy and recommended phase 2 dose of both Teclistamab and Talquetamab were ascertained, the next step was, can we combine them with other myeloma treatments and see if, we get, better clinical activity. So the TRIM-2 study is looking at the subcutaneous daratumumab administered with, Talquetamab as well as with Teclistamab. And there is combination study of adding pomalidomide, to those combinations as well. So we haven't heard about the POM combos, but we know the sub-Q dara combinations. They have been reported out by my colleagues, Drs. Krishnan, Dr. Jayachari, Drs. Vanderdonk. Each of them have presented these data at different congresses over the past, 6 or 8 months. And I've participated in those studies as well. The overall response rate with each of those combinations is about 80-odd%. Which is remarkable. These, because most of these patients were daratumumab refractory in a previous line of treatment. So why are we seeing a higher response rate compared to what we would expect with Teclistamab and Talquetamab? Daratumumab also is inhibitory to regulatory immune suppressive cells by virtue of blocking CD38. So it's really acting as an immune modulator and enhancing the effects of these. And that's what I feel that's probably the reason why we're seeing this activity. But those are very high response rates, right? With both Teclistamab and Talquetamab, I've shared that the responses are in the 60%, but you're seeing a higher percentage. So very encouraging. What does this mean? It means that, we can potentially, use this combination in earlier lines of treatment, even for newly diagnosed patients. It would be really cool to have a non-chemotherapy approach just using antibodies.
Right now, is it still in that triple class refractory population?
Yes, yes, but there are clinical trials in earlier relapse and even in the newly diagnosed setting being planned. They're not in the public domain yet, but they're planned, and we're probably going to have these bispecifics examined for newly diagnosed patients soon.
Good. Yes, especially maybe that might be something to consider for like the high risk group, which is really still quite an unmet need here in myeloma. Let's talk about some of the earlier lines of treatment. I think there was a trial that was presented that talked about RVD plus or minus elotuzumab. Was that correct? Or?
Yeah, so this was an update from the SWOG 1211 trial. So just to give you a context, of about high-risk patients in general. So myeloma is a very heterogeneous disease. Biologically, we have, 8 or 9 different subgroups, depending on how, you classify it on a molecular basis. From a clinical behavior standpoint, the way that we, classify myeloma is patients who are at a higher risk of relapsing early after getting frontline treatment compared to those who have standard risk, in which we in those patients we don't expect the myeloma to relapse early. So high risk is defined by certain clinical features such as circulating plasma cells or extramedullary myeloma, or certain karyotypic abnormalities like translocation 414, 1416, 1420, or deletion 17p, as well as amplification of 1q21. So, the SWOG 1211 was the first clinical trial that I had the privilege of, designing and running as when I was a junior faculty within the US cooperative group setting focused on high-risk myeloma. So if you look at all the clinical trials, the Maya, all the major studies that have been reported, they take all comers, any myeloma patient can participate. And what we recognized as clinicians is high-risk patients don't do well. So the idea of the SWOG 1211 was, let's design a trial just for high-risk patients and see if we can make a difference for those patients. So that was the first effort. The idea was giving RVD for induction and then continuing RVD as maintenance. So the dual, PI IMiD maintenance strategy. And then in the experimental arm, elotuzumab was added on to that same RVD, backbone, both in induction as well as maintenance. It was a randomized phase 2 study, and the study did not show a benefit of elotuzumab adding anything to RVD. However, the trial showed better PFS than our historic control. So the median PFS for just the RVD arm was about 34 months. And our assumption was that the median PFS for those patients would be about 26 months. And that assumption was based on patients getting tandem transplants. So, this was from the total therapy time. So the RVD arm actually performed better than the older therapies. So, so those are kind of, SWOG 1211 set up a benchmark for other high-risk trials to follow. And, at the EHA meeting, at the ASCO meeting, we heard about a UK study looking at KRD data for high-risk patients. There's an IFM trial. And there's also a KRD isatuximab study from the German myeloma group. These are smaller single-arm studies that are trying to do the same thing, do enrichment design trials for high-risk patients. So, I wanted to give, the whole context of why that study is important. The results by themselves for SWOG 1211, it's considered a negative study because ELO didn't add anything, but then it set up a very important benchmark for other studies to improve upon.
That's good to know. I really didn't know that background. And I'm glad to hear that more and more studies are just targeting that high-risk myeloma population. It's so important, to figure out how can we control that disease early on, in that group of people. How about the determination trial? I hear so much about it. I still haven't quite figured out what the results mean. Can you try to clarify that in my mind?
I can. It's very interesting. So that it's a positive study, right? So the primary endpoint for this particular trial was, showing PFS benefit between doing an early transplant or delaying it at the time of, the patient's first relapse. So if, so that was the primary endpoint, and there is a 21-month benefit, in favor of doing transplant early for that young myeloma patient. The median age on this study for patients enrolled was 55 years. So this is a young patient population, right? This is a sister trial to the IFM 2009 study that had already been reported out about 5 years ago. The response rates, the depth of response in terms of MRD negativity, and sustained MRD negativity appears to be, higher in the transplant arm as well. The study has a median follow-up of about 70 months. And at that point, median OS has not been reached in either of the arms. So some investigators, the way that they're framing that is incorrect. They're saying, oh, there's no OS difference. No, that's not how you read the statistics. You don't say, there's no OS different at a time point when the median OS has not been reached in both arms, because there's not enough follow-up. If that was the case, we would be saying the same thing about the Maya trial, the Alcyone trial, the Cassiopeia trial. All of those trials had no OS benefit when they were reported out because the median OS had not been reached in those arms. And the other important thing that we see is there is no difference between the two arms between second primary malignancies as well. The kind of second primary malignancies that people get in one arm over the other are different, but the overall percentage is about 10% that was reported out. So again, that's my take on the study. It really doesn't add any new information. We already knew the IFM 2009 study data. What it does augment is the use of LEN maintenance, because the IFM 2009 study stopped maintenance after a year, but the determination trial continued maintenance for everyone until relapse, progression, or intolerance. So the study really doesn't add a lot more to what we already practice. And I hope that this is the last trial that we're doing where we're thinking about early versus late transplant. Because that is an old question. Now the real questions, the new questions should be, what about CAR-Ts? What about bispecifics? I hope so we're designing trials with that question in mind. I think there's a lot of hype around determination, but really it's hype. I don't think it adds much to what we are doing right now.
Right. And for those watching, early transplant means transplant is part of your induction therapy. Late transplant means at first relapse. Is that what the trial was talking about?
Yeah, and since both arms did pretty good up, in terms of median PFS. So median PFS if you got transplant was around 68 months. Median PFS if you just got RVD and delayed transplant was about 47 months. So there was a 21-month difference in favor of early transplants, and the follow-up is short, on the study. So you're not going to have a lot of people, even in the RVD arm, relapsing to get their transplant. So and myeloma patients are doing so well today. The median OS is at least 10 plus years, right? So you can't just say after a 4 or 5 year follow-up that we're seeing OS differences. So I'm hoping that by the time we get the determination OS data, it's going to be 10 years from now, okay? Because our patients are going to be doing so well that we're going to be looking at this historic data and saying, it was a good question at that time, but probably not relevant to us by the time that reads out.
Exactly. In 10 years, who knows what our induction therapy is going to look like? Much better, I'm thinking. What about the endurance trial? Can you talk a little bit about what that was?
So the endurance trial ran side by side to the SWOG 1211 study, and it was asking an induction question for standard risk patients. So it was asking the question, is KRD better or RVD better for induction? So, endurance was a randomized phase 3 study. Half of the patients got carfilzomib with Lendex as maintenance, as induction. The other half got, VRD or bortezomib with Lendex as induction for 8 cycles. And then everyone got Len as maintenance. This study enrolled standard risk patients because high risk patients were going on the SWOG 1211. But translocation 414 patients were enrolled on this study as well. So that trial read out showing no differences between PFS on either arm of the study. Essentially for standard risk patients, it established RVD as the right treatment choice, just like the SWOG 777 trial had shared with us. The other interesting thing about the median PFS of RVD, on the endurance trial is the median PFS was around 34 months. So again, the SWOG 1211 with the PI IMiD maintenance showed the same thing. So my colleague Shaji Kumar presents this slide, saying what makes the difference in high-risk patients is the dual maintenance. If you look at, the endurance trial and SWOG 1211, together because both of these were enrolling at the same time.
So dual maintenance is what's happening now in high risk?
For high risk
basically?
patients.
Okay. Anything else that we should be talking about? Anything else new and exciting?
Oh, wow. I mean, there is, a lot of, there are a lot of new trials that are kind of in the works where, there's the CARTITUDE-5 study, which is looking at patients getting RVD as part of induction. So these are patients who, either are transplant ineligible or they're deferring their stem cell transplant to first relapse. Everyone gets RVD as part of induction treatment, but then half of the patients get randomized to getting CAR T-cell therapy. Other half, they move on to their maintenance treatment. And the people who get the CAR T-cell therapy, they don't get any maintenance. So that's a really cool study. We're hoping that it can provide a good answer for those patients. The second trial is actually, looking at transplant-eligible patients. So everyone gets, the quadruplet regimen of daratumumab with RVD, and then patients get randomized to getting either a transplant or CAR T-cell therapy. So that's a very exciting study where we're hoping we can get a positive answer for our patients. High dose melphalan and transplants have been a part of myeloma treatment for a long time, but if we can replace them with another cellular therapy approach, which is more specific to the myeloma cells, I would really welcome that.
I think I would welcome that too. And that brought another question to my mind. When you talked about transplant-ineligible patients, there's that group of patients that really are not eligible for transplant for whatever reason. Would those group of patients not be eligible for CAR T-cell therapy, or would they be possibly eligible? I know you have to look at them individually, but as a group, is it just like you're not eligible for this, so you're not eligible for that, or?
So I think we are a little bit more lenient in CAR T-cell therapy eligibility compared with stem cell transplantation because patients are not getting the same level of myelosuppression with the high dose chemotherapy like a stem cell transplant. So there might be patients who are in their mid 70s or even late 70s who are able to get CAR T-cell therapy that we would not necessarily pick for a transplantation. So I think that there are we're thinking about CAR T-cell therapy in a more lenient way because of the safety profile.
That's good to hear. So, wow, we talked about a lot today, and it's just amazing, all these new therapies. I know when I was diagnosed back in 2008, I think most of the things we talked about weren't FDA approved except Revlimid and Velcade. All the rest have been around. So when you see a newly diagnosed patient or even an older patient who may be relapsed refractory, how do you provide them with hope with all these new treatments? What are your words of wisdom to these patients?
I give them the context. I tell them exactly what you said, Cindy, and I tell them about the time when I was starting in the field in the early 2000s and how the outcomes were for myeloma patients, and I walk them through how things have improved, what our goals and expectations are today for, for our patients and the treatment options we have. So I provide them with measured hope. We do recognize that myeloma is still not, a disease that we can, cure, but we can control it for a long duration of time. And even for patients who have high risk myeloma, we're thinking about things in a different way. And trying to be proactive in giving different strategies for those patients rather than just treating them in a vanilla, one size fit all kind of a way. So, I provide, that context to patients, and then, it's really about individualizing treatments based on patient preference as well. this is really a partnership. Our job is to provide patients with as much information as they can, gather to make good informed decisions.
Do you think we'll be curing any myeloma patients anytime in the future? And what do you think that would look like?
So I would say yes. I think, this is something that I've shared, on other forums as well. I do feel that if we can, get patients to undetectable levels of myeloma over time. So, there's this, term called MRD, or minimal residual disease negativity. If we're able to get patients to MRD negativity and maintain them in that MRD negativity for a certain period of time, and then stop therapy at that time, then we can, functionally cure, majority of patients. Myeloma is a disease that tends to occur later in life. There are other competing health issues that patients have. So a true cure, cannot be determined when, in that kind of a situation, compared to someone who says, someone who was 20 and they got diagnosed with a cancer and they die of a heart attack at 90. You can say, okay, this person was cured from a cancer. You can't say that about, a myeloma patient who's getting diagnosed in their 6 or 7 decade of life. But, if you can, kick myeloma down to undetectable levels, stop treatment, and patients, continue to thrive for years, and the cancer doesn't come back, that's, in my mind, functional cure. Yes, my answer is yes, we're going to get our patients there. The key would be finding the right recipe to get patients to that sustained MRD negativity before stopping.
Right. And now we have so many ingredients for that recipe that I'm sure we're going to find it soon, I'm thinking. Thank you, Dr. Usmani, for a wonderful conversation. We learned so much, and we have so much to be hopeful for with all these new treatments. I really do see a cure in the near future. So once again, this is Cindy with The Patient Story. Thank you for watching.